Thank you for the opportunity. I'm really excited to discuss our paper that was recently published in Nature Communications entitled Integrative Multiomics Analysis Uncovers Tumor-Immune-Gut Axis Influencing Immunotherapy Outcomes in Ovarian Cancer. And this was essentially a corollary study for a clinical trial where we combined pembrolizumab, cyclophosphamide, and bevacizumab in treatment-refractory ovarian cancer patients. And just in terms of disclosures, I have no financial disclosures, but I'd be remiss not to mention that Merck did support the clinical trial and some of the translational research shown here through their competitive MISP program, which was obtained by Dr. Amesha Siros, who's the chair of gynecologic oncology at Roswell Park. And this trial and this translational research would not be possible without her and her group. So why exactly are we even studying ovarian cancer? It's the #1 cause of gynecologic cancer deaths and the #5 cause of cancer-related death in women. But more strikingly, despite a 5-year survival rate of 49% and a 10-year survival rate of 35%, there are no early detection tests for ovarian cancer. And there's this common misconception that a Pap test will detect ovarian cancer, when in fact it will not. And this leads to the vast majority of women presenting with advanced disease. And in fact, only 16% of cases are diagnosed in the early stages. And this is largely due to a very broad symptomology like bloating and constipation, urgent urination, difficulty eating, fatigue, things that can be attributed to many other disease states and often are attributed to many other diseases before ovarian cancer is ever considered. And so over recent decades, while the 5-year overall survival of women with ovarian cancer has improved, this is largely due to more treatment lines rather than better first-line therapies. So we just continue to treat and treat and treat as ovarian cancer becomes refractory. And platinum and paclitaxel, which are highly toxic, are still the main drugs that have been in standard use for over 20 years for these patients. Treatment of platinum-resistant ovarian cancer is really the crux of what we are studying, as it continues to be clinically challenging. It has really poor outcomes, and the median progression-free survival benefits are modest at best. And in fact, response rates to second-line cytotoxic chemotherapy is only about 15 to 25%, with an abysmal median progression-free survival of 3.4 months. And so, like many other disease states, immunotherapy and the immune state of cancer is really being explored as a novel aspect or a novel treatment modality. And so intratumoral T cells or tumor-infiltrating lymphocytes have been shown as a prognostic factor in ovarian cancer. Many of you are probably familiar with the concept of a hot TME versus a cold TME, where a hot tumor microenvironment is highly immune inflamed. We have a large number of tumor-infiltrating lymphocytes, as shown here in this IHC. A cold tumor microenvironment includes immune exclusion, or otherwise phrased as immune ignorance, where we see a total lack of tumor-infiltrating lymphocytes. And interestingly, in this study, they did find at 96 months, for those individuals that had what we call a hot TME with that large infiltrating tumor lymphocyte population, there was a 50% population in remission, and we saw significant improvements in progression-free survival. Further, in terms of overall survival between months 96 and 132, we did see an increase in about 60% that were still alive. So TILs as a prognostic marker does make sense for us to pursue. However, interestingly, when we look at the efficacy of single-agent anti-PD-1 and PD-L1 therapies in ovarian cancer, some of the largest studies, including this phase 2 recurrent ovarian cancer study, which included 376 patients, had really abysmal outcomes with just a progression-free survival of 2.1 months. So very limited benefit of single-agent anti-PD-1. Another treatment modality that has been of interest is Avastin or bevacizumab, which is an anti-angiogenic. And when combined with chemotherapy, we did note that the length of time that women lived without their tumors doubled. So it went from about 3.4 months to 6.8 months when Avastin was used in combination with chemotherapy. Additionally, we saw an increase in the number of women with tumor shrinkage. So it went from 13% to 28%. However, recurrence rates were still high and progression-free survival is still limited in these populations. And so that brings us to the phase 2 clinical trial that Dr. Amesha Siro started at our institute, which combines pembrolizumab with bevacizumab and oral metronomic cyclophosphamide, which is typically used as a maintenance therapy. And in this study, we had several treatment refractory patients where at baseline and on treatment, we collected tumor biopsy, serum, as well as stool for the microbiome. They had several images done throughout the breadth of the study, and we very specifically looked at progression-free survival, as well as objective response rates, overall survival, and toxicity and quality of life. What I'll be discussing today were our exploratory endpoints, where we looked for changes in the TME, changes in circulation, the stool microbiome, and metabolite composition. Now, interestingly, with this combination study, we were able to extend median progression-free survival to 10.8 months, which is pretty good in this group of patients. But perhaps more interesting than that is we were able to designate patients that had either durable clinical response or durable clinical benefit, who not only obtained median progression-free survival of at least 10.8 months, but also saw a reduction in tumor size of greater than 50%, and those with limited clinical benefit who did not see that same reduction in tumor size and had less progression-free survival than that median 10.8 months. And when we compared our durable clinical benefit with our limited clinical benefit individuals, we saw massive differences between them. And in fact, the median progression-free survival of our durable clinical benefit patients was 20.2 months, which is really unheard of in this population of individuals, whereas those with limited clinical benefit observed about a 5.72-month median progression-free survival. And this really prompted us to want to understand what delineates those patients with durable clinical benefit, which I will refer to as DCB from here on out, as compared to those with a limited clinical benefit. And so first we started with our punch biopsies, looking at the tumor RNA sequencing. So this is bulk RNA sequencing from those tumor biopsies. And not surprisingly, when we compared at baseline and on treatment, our DCB and our LCB patients, we saw really strong immune signatures. Regardless of baseline or on treatment, those with a durable clinical benefit observed enhanced T lymphocyte and T cell activation, CD40 pathway engagement, B cell proliferation, antigen presentation, and cytokine signaling. Whereas those that observed a limited clinical benefit were really enriched for things like positive regulation of synapse assembly and neuronal differentiation, as well as broad oncogenic signaling like MYC targeting. And further, when we did an immune deconvolution, we saw, not surprisingly, that in our DCB patients, regardless of whether it was at baseline or on treatment, there was just larger portions of infiltrating immune cell signatures based on this deconvolution algorithm as compared to our limited clinical benefit patients. But perhaps more interestingly, from my perspective, given my career has been focused on metabolism, we did see transcriptional dysregulation of metabolic transcripts. And so to this end, we utilized the bulk RNA sequencing and applied Metaphor, which is an algorithm I developed back in 2018 to assess metabolic dysregulation using transcriptomic level data, whether it's RNA sequencing or microarray, and it can tell you which of 114 metabolic pathways are most highly dysregulated between any two conditions. And when we looked very specifically at the baseline LCB versus DCB patient populations, we found enrichment of pathways like glycerophospholipid metabolism, tryptophan metabolism, and its connected kynurenine metabolic pathway, as well as ether lipid metabolism and linoleic acid metabolism. Further, when we looked on treatment, we saw many of the same pathways, including tryptophan and glycerophospholipid metabolism. But beyond that, we also saw enrichment of pathways associated with drug metabolism and metabolism of xenobiotics, which may explain this differential response to therapy. But the question really becomes, what is happening metabolically? And that's because we already know that even though the algorithm has been shown to properly and appropriately predict metabolic pathways of importance based on transcriptional data, the relationship between transcripts and metabolites isn't necessarily linear, especially given things like post-transcriptional and post-translational modifications, and also the fact that the metabolome is really your phenotypic endpoint readout. It's not only what's happening endogenously, but also it is a result of some of those exogenous effects like diet and exercise and even the microbiome portions. And so, unfortunately, in a study like this, we're very limited by sample type in these patient populations. When patients present with latent stage disease, like all of the patients that were included on this trial, they do not typically undergo any sort of surgical resection, which means we only really have a few core biopsies, which were used for RNA-seq, digital spatial profiling, and whole exome sequencing. What we did have, though, was serum. And this to us was more of a circulating metabolome, which was available not only for feeding the tumor, but also for feeding the circulating immune cells, which would eventually potentially be trafficked to said tumor. And interestingly, when we looked at our DCB and LCB populations based on the Biocrates MXP Quant 500 kit, which had all of those stable metabolites, we did in fact find very specific lipid and amino acid components that were highly associated with durable clinical benefit. One of the largest of which being 3-IAA, which is involved in the tryptophan and kynurenine metabolic pathway, which was highly enriched in our tumor RNA-seq, but is also highly bacterial in nature. And this prompted us to utilize the fecal samples to also look at the excreted metabolome and how potentially the gut may be impacting therapeutic response, especially given the resurgence of papers that have recently come out of individuals like Jen Vargo's group, which assert that the microbiome and the gut axis might actually impact immunotherapy response. And so we very specifically looked at the fecal metabolome in our LCB and DCB patients, both at baseline and on treatment. We found similarities with treatment. So for example, fatty acid 12O was up with treatment in both our LCB and DCB populations, regardless of response. And so this is more than likely attributed to the therapeutic regimen. But we also saw differences in terms of things like triacylglycerides, cholesterases, phosphatidylcholines, and these extended to on-treatment comparisons of LCB and DCB, as well as baseline DCB and LCB patient populations, very specifically focusing on those metabolites which were altered with treatment, very specifically in our DCB group. We found things like hypoxanthine, which is related to purines, as well as several amino acids like glutamine and serine, and acylcarnitines. And so what this really told us is that the fecal metabolome is demonstrating alterations in key lipids and microbial metabolites that are highly correlated with altered response to combination therapy in our responder populations. But this prompts the question, where exactly are those changes coming from? So utilizing the DNA and RNA that we had already extracted based on our core biopsies, as well as our fecal matter, we looked very specifically at the gut microbiome diversity that was linked with the varied ovarian cancer response. And we were able to identify several bacterial species that were highly associated with alterations very specifically in our durable clinical benefactors, some of which have been highly associated with immunotherapy response in the literature, like Butyrate producens, which is a butyrate-producing bacteria, as well as some that are highly associated with fecal samples in general, like Peptococcus and Raminococcus. But more interestingly, we were also able to identify novel microbes like Dielma fastidiosa that maybe hadn't been associated with therapeutic response in these populations previously. And interestingly, when we looked very specifically at each of these gut microbes, we found those that were increased in our DCB patients as compared to our LCB patients, regardless of time point. So regardless of whether it was at baseline or on treatment, like Butyrate producens, we see increases in our DCB as compared to our LCB regardless. We were also able to identify bacterial species that were only increased at baseline but depleted with treatment. And we were able to identify bacterial species increased with treatment in patients with DCB only, which are really the ones that we're focused on. But more so when we looked very agnostically at each of these bacterial populations, we were able to identify those species like this Butyrichy producens that when we divided their OTUs, their operational taxonomic units, by median expression, there was in fact a survival benefit in our patient population based on solely the expression of those particular bacterial species. And this to us was indicative of a gut immune metabolic interaction in these patients, given that each of these different components were highly associated with therapeutic response. And interestingly, as I said, we also utilized the DNA RNA that had already been abstracted to look very specifically at the tumor microbiome, which showed more limited alterations. And this is indicated by the fact that our fecal microbiome was able to produce well over 800 microbial species, whereas our tumor microbiome, we had just barely over 300 species, and only 38 actually overlapped between the two. However, we did involve some functional redundancy testing. So even if the bacterial species itself was different, were they functionally resulting in some of the same consequences? And interestingly, what we found in terms of the tumor microbiome was that when we compared baseline DCB versus baseline LCB populations, we once again were enriching for things like amino acid metabolism, as well as inflammation and infection, which tells us that we're eliciting some sort of immune response, but also some sort of interplay with the metabolic milieu as well. And so given the enrichment of both the inflammatory and metabolic components as related to the microbial signatures, we really wanted to start to explore all three of these components together. And so the first thing that we did was we looked at the most highly dysregulated metabolites like DHA and serine, TMAO, as well as the most dysregulated microbial populations like Peptococcus, intestinale, Dielma fastidiosa, and we wanted to correlate them with these immune deconvolution signatures that resulted from the RNA-seq. And we were able to pinpoint very specific interactions like glutamine being highly positively correlated with CD8 positive T cells, which was also seen in bacterial species like Dielma fastidiosa. But again, like I said, we don't necessarily have that linear relationship between our transcripts and our metabolites. And so this warranted looking at each of these independently and finding things that they enriched for together. So taking into account that we had already done this metabolite enrichment analysis essentially from our RNA-seq using Metaphor, we also did a metabolomics analysis where we enriched the metabolomics from the stool samples, and we found glycerophospholipid metabolism, fatty acid elongation, fatty acid biosynthesis, and amino acid biosynthesis. So all things that were highly consistent with some of the things that were enriched in our tumors. Additionally, we did a 16S pathway prediction, and we enriched for, once again, amino acids like glutamine degradation and tryptophan degradation, as well as our lipid components like fatty acid activation and diacylglyceride biosynthesis. And further, when we overlapped that metabolic enrichment from our RNA sequencing with the 16S enrichment and the metabolomics enrichment, there were three common pathways: tryptophan kynurenine metabolism, glycerophospholipid metabolism, and amino acids, specifically glutamine and glutamate or urea metabolism. And so these were really the pathways that we focused in on as potentially being altered or having the ability to alter response to our therapeutic regimen. And so we really wanted to look at this co-enrichment of this differential analysis from the tumor transcriptome, gut microbiome, and fecal metabolome, which did reveal the lipid and amino acid pathways of interest. But we wanted to really delve into which showed the most important relationships. So we created a co-expression network, essentially, to identify, based on the immune deconvolution data, which immune pathways were most highly associated with which microbes, which are indicated by these orange ellipses, and metabolites, which are indicated by the green rectangles. And so we were able to really identify patterns like CD8 positive T cells were highly positively correlated with Dielma fastidiosa, which was impacted by things like serine, TMAO, and glutamine, whereas there was a negative relationship with indoles and tryptophan metabolism, which is not all that surprising given what we know about tryptophan and kynurenine metabolism and their impact on immune cell function. And so beyond that, because again, this linear relationship doesn't exist, we really started to build these pathways, layering in all of the different levels of multi-omics data so that we could pinpoint what we call biological points of convergence. And to us, these are points where even though we're looking at snapshots in time of all of these different multi-omics related data, when different levels of that data are pointing to very similar biological phenotypes, these are perhaps things that we can target and leverage. And so, for example, we had the tumor transcriptome, and since the only level of data that we're showing here is the transcriptomics, we did see decreases in IDO1 and kynurenine, which would allow us to predict potential impacts on tryptophan, kynurenine, and anthranilate, which we are now functionally testing in a system in the lab. However, when we're talking very specifically about the microbiome and the fecal microbiome, we were able to integrate not only those fecal microbiota and the transcripts associated with those microbiota, but also the fecal metabolome. And here we showed that there were increases in tryptophan with subsequent decreases in kynurenine and indole. And these corresponded with decreases in microbiota transcripts of kind B, VAS1, and ISS1. And all of this makes sense from a biological standpoint in that you are not shunting tryptophan towards the generation of indole or kynurenine in our responding patients, which means that we're not getting that overwhelming reprogramming of those CD8 positive T cells that we know are associated with indole and kynurenine in these CD8 positive T cells. So there's not as much availability of the indoles and kynurenine to actually functionally remodel those T cells. And once again, we're now actually testing this in the lab. And so really, we're starting to delve into this layering and modeling approach to better understand the crosstalk between the tumor, the gut microbiome, and the fecal metabolome to reveal those biologic points of convergence. And this has allowed us to really generate this overall model of what we think is happening, where when we treat patients with pembrolizumab, bevacizumab, and cyclophosphamide, we're getting this durable clinical benefit that's associated with an abundance of specific beneficial bacteria, leading to beneficial microbiome-derived metabolites like TMAO, serine, and glutamine, which lead to an inflamed tumor microenvironment on treatment that are associated with increased B cells, CD8 positive T cells, CD4 positive T cells, DCs, and even TLS signatures. Whereas in our limited clinical benefit patients, we're seeing increases in what we call non-beneficial bacteria or harmful bacteria that are associated with things like DHA and indole and betaine, creating a cold or immune desert in those particular patients associated with increases in potentially neutrophils, leaving them less susceptible to the utilization of this combination pembrolizumab, bevacizumab, and cyclophosphamide. And so we're now getting more into the molecular mechanisms that are behind this, and we're hoping that'll be a paper coming out soon. But overall, pembrolizumab is combined with bevacizumab and oral cyclophosphamide is now an active regimen that's been included in NCCN guidelines thanks to this paper and Dr. Amesha and the results of this trial. We found differences in T and B cell tumor infiltration and function both at baseline and on treatment based on tumor transcriptomics and digital spatial profiling, which I didn't show, as well as some of our whole exome sequencing of the tumor and stromal regions. The metabolic assessment of the tumor transcriptome, fecal microbiome, and fecal metabolome revealed glycerophospholipid, tryptophan, and kynurenine metabolic dysregulation underlie therapeutic response. And we do think that the gut immune metabolic axis may impact therapeutic response in ovarian cancer patients on this particular regimen. But we do know that future studies with other therapeutic regimen and sample collection, especially those that include our ability to collect specifically for the metabolome, are necessary. And at this point, I'll very quickly acknowledge this study would not have been possible without Dr. Amesha, again, the head of our gynecologic oncology department here at Roswell. It's her clinical trial, and she did a phenomenal job. We would be remiss not to mention all of the patients on the clinical trial, who these biospecimens came from, the entirety of the gynecologic oncology team, including all of the doctors and support staff, including nurses, PAs, and clinical trial. individuals who were involved in this are biostatistics and bioinformatics team, including Mark Long, who's my co-first author on the paper, as well as Bashan and Eduardo, who are master's level biostatisticians, the entirety of the gynecologic translational research team, the genomics facility, and my group in the bioanalytics, metabolomics, and pharmacokinetics facility, as well as our funding sources.